the webinar.
Thank you, Dai, and hello everyone. It's my pleasure to welcome you to another talk in the 2025 series of our Econ-Omics talks. My name is Ilias Gouranitis, and I'm a health economist at the University of Melbourne. Today, and on behalf of our conveners, I'm delighted to introduce our speakers, Professor Zanfina Ademi and Dr. Padraig Dixon. So they will talk to us about the application of Mendelian randomization in health economics and how we can bridge genomics and cost-effectiveness. Now, Zanfina is a professor of health economics within the Faculty of Pharmacy and Pharmaceutical Science at Monash University, where she leads the Health Economics and Policy Evaluation Research Group. She has worked in Australia, Finland, and Switzerland, leading impactful research, particularly in the context of disease prevention and management, population genomic screening, and the development of productivity-adjusted life years. Dr. Padraig Dixon is a senior health economist at the University of Oxford, and his research explores the economic implication of genetic data with a focus on causal inference, screening, and evaluating the cost-effectiveness of stratified therapies. His work spans multiple disease areas, including cancer, cardiovascular diseases, and dementia, as well as traits, behaviors, and interventions such as pharmacogenetics. Well, it's a pleasure to have you both here. We'll stop sharing, and it is a convention here within the economics talk to ask you a question about your career that will benefit particularly our IHEA group. So the question for you is, reflecting upon your career, what advice would you give to our ICRs to help them grow into independent and impactful researchers? I'll start with you, Zanfina.
Thanks, Ilias. Thanks for the invitation. It's great to be here and great to see so many of you attending the talk. So in terms of advice, so I've been very fortunate to foster collaboration across academia, non-governmental organization, government, industry, and consumer-led organization, including serving an advisory board. So I feel like these experiences have allowed me to learn from a wide range of perspectives and have been an invaluable part of my professional growth. So if we want to solve bigger problems in healthcare or complex problems, we must draw on diverse perspective and work together. So this is my advice. But also, you should focus on why. What matters most isn't what you do if you build a great economic model, but you should focus on why you do it and why you are motivated to wake up every day in the morning to deliver the right treatment to the right person at the right time, and obviously balancing efficiency and equity. I asked one of my PhD students that I mentored, what would an ECR like to know, what extra activities actually help your career progress. I mean, for me, my advice will be cross-train in purpose, because I'm myself cross-trained in purpose as a pharmacist, epidemiologist, and then health economist. So during my time as a master student in public health and a PhD in health economics, I actually took a lot of classes that were, for example, neuroscience, neuroplasticity, topics completely out of my lane, but they changed the way how I think. And I also wandered through museums and art centers, and I think art and architecture helped me reframe problems and see research questions from a different perspective, not just from a health economic perspective. So this is my broad advice to you. As much as you get focused or get exposed to different perspective, I feel like you would have a broader understanding of a complex problem in healthcare. Thank you.
Thanks so much, Zanfina. Padraig, what are your thoughts?
Yeah, thanks, Ilias. I guess agreeing with everything Zanfina said, and two other specific reflections. One is, I think the importance of viewing your PhD in early career as a type of apprenticeship, and that means you're wanting to learn. And I think being in an environment where you're challenged and where you're working with people at the research frontier is really important, because that will lay the foundations for everything that you subsequently build on. I think also saying yes to things that challenge you. I think it's easy to stay in a comfort zone, but saying yes to different topics, to different techniques, partly so you develop skills, but partly because you'll develop networks, you'll be exposed to different ideas, and you never know really where those things go. I think there are very few people who will have had a completely linear path in academia where you start in a particular topic and then you never deviate or diverge from that topic. I think exposing yourself to high quality research environments, exposing yourself to challenges, those are all things that will lay a powerful foundation for what comes after in your career.
That's great advice. Thank you very much, both. I'll hand over to you, Zanfina, now. Your talk, I think, is about 20 minutes, and then Padraig will follow with another 20 minutes presentation. Then we'll wrap it up with some Q&A and some of other events that the SIG will be doing. So over to you, Zanfina.
Thank you. So can you see it in a presenter mode?
Yes.
Okay, great. Just before I start, some conflict of interest. I'm a member of the Economics subcommittee, and I sit in a lot of international task forces, and also funding for this project from NHMRC Australia. So what I want to talk today is just to give you a brief overview, what is Mendelian randomization, and I'm sure Padraig is going to be able to even explain even better than I. My focus is going to be cardiovascular disease, but what we want to achieve with this talk today is understand the role of Mendelian randomization in health economics and how we can use it. And my focus is mostly on using Mendelian randomization as evidence to inform prevention strategies. And then I will give you a step-by-step guide on how we have estimated the causal lifetime risk in cardiovascular disease using Mendelian randomization studies, and then some case studies where we have applied this method. So why Mendelian randomization? So knowing if a risk factor causes an outcome help us understand if addressing that risk factor could prevent the outcomes. So we know that with observational epidemiology, we have always issues with causality. We tend to measure correlation, but correlation doesn't mean causation. So there is always residual confounding that we deal with in observation epidemiology. Then we solve this issue by doing randomized clinical trials. So we want to intervene by knowing a causal relationship between a risk factor and outcome. But we know that clinical trials are very expensive, and that's why Mendelian randomizations. So what is Mendelian randomization? Mendelian randomization exploits the fact that alleles are segregated randomly during the meiosis. So those, when people are grouped by their genotypes at the population level, it can be assumed all confounders are equal between the groups. In this example, you can see that you have two different alleles. One of these alleles is associated with higher risk factor level, and then consequently, this higher risk factor level leads to your higher risk of developing an outcome. So both Mendelian randomization and randomized clinical trials are two study designs that afford randomized evidence in human biological and medical research. So both capitalize the power of randomization to provide unconfounded estimate of causal effect. Randomized clinical trial in one way, randomly allocating people to control an intervention, whereas Mendelian randomization uses genetic variants to look at the relationship between risk factors and outcomes. But when we look at Mendelian randomization, MER relies on several important assumptions, and this is very important to highlight that the variant is associated directly with the exposure. So this is one first assumption that we have to be aware of. The second assumption is that variant is not associated with the outcome via the confounding pathway. And the third one is that the variant or allele allocation do not affect the outcome directly, only indirectly via the exposure. So these are the three most important assumptions that you have to consider when you do the Mendelian randomization studies. So the role of Mendelian randomization in health economics. So Mendelian randomization has been used for ages in epidemiology, but it's been a couple of years that there have been several publications highlighting the implications of Mendelian randomization, how they can be used in health economics. And I believe that Mendelian randomization offers solutions when randomized clinical trials are impractical for delivering causal effects in health economic models. It helps address limitation of observational studies in estimating the risk factor modification. And this method, as you've seen by my previous slides, combines disease pathophysiology knowledge and epidemiological evidence for causal inference from observational data, and is useful for designing models that incorporate long-term effect of risk factors on a disease. So there are two ways how you can potentially use a Mendelian randomization in health economics. One way is to look at the genetic variants by looking at the single nucleotide polymorphism related to a risk factor. In this exercise, I'm showing you that we've used the UK Biobank data, which is one of the largest data set in the world that has data on genomics. And based on this information, you can look at the relationship of alleles with a risk factor. So you have the SNPs here, which are related to systolic blood pressure. And we use Mendelian randomization to look at the relationship between those SNPs in relation to quality of life. So what does this method do? It looks at the relationship that for every millimole reduction or increase in systolic blood pressure, how is your quality of life? It's going to change over time. In a similar fashion, you can actually look at those SNPs, systolic blood pressure. Instead of looking quality of life, you can look at costs. So option 2 is microsimulation and health economic modeling. And this is what I'm going to be focusing today mostly. So this is using estimates from published studies to design a health economic model. And the reasoning why we do this is that short-term randomized clinical trials provide estimates. They might underestimate the benefits of long-term prevention. And that's why we use Mendelian randomization studies, because they provide a cumulative effect of major risk factors like cholesterol, systolic blood pressure, smoking pack years, and diabetes time. And this is it's not captured when you do a randomized clinical trial. And because of this, if you don't have a long-term clinical trial, this may lead to misallocation of resources. So that's why we think that MR studies will provide information on concept of risk factor burden or cumulative burden and economic evaluation over time. And also this is important when we want to think of economic value of prevention. So if Mendelian randomization can provide such information, then we will be able to show that prevention strategies go beyond healthcare savings, so create positive impact across multiple sectors such as labor, education, and environment. And prevention is often expected to save healthcare costs. In many cases, it leads to additional long-term spending from increased longevity. Delaying healthcare costs does not reduce the value of prevention. What matters is whether the overall societal benefits outweigh the costs. And we actually have to capture that outcomes that matters to society, not only to healthcare. And also we have to diversify our evidence for prevention to inform decision-making. And that's why Mendelian randomization as a real-world evidence plays a critical role for prevention strategies. With regards to cardiovascular disease, we know that the current prevention approaches are mostly initiated late in life. And majority of these clinical decision tools bias decision-making via short-term decision-making, meaning that we only look at your risk factor at one point in time. We don't look at over your lifetime. And majority of these risk scores that we have produced or use them in clinical practice are influenced by age and gender. So modifiable risk factors play a very little role. So this is a way how I have approached stages of using Mendelian randomization evidence for health economic modeling. So here I'm using evidence that others have published and generated with regards to risk factors. And I'm combining this by using data from UK Biobank. So the first thing that you have to know, you have to select your population and intervention, of course. But then you have to simulate the risk factor trajectories, and this risk factor trajectories have to be over lifetime. So we have published data to show that risk factor trajectories for individuals from age 0 to age 85. And then you have to convert calculate transition probabilities by converting the causal estimate from MR into effect of an outcome. And as I pointed out, this is by unit time. And then you can design a micro-simulation model and perform the health economic analysis. So in our case, we've selected the population from UK Biobank to simulate the impact of risk factors trajectories over time. So the first step was to identify those risk factors that have an impact on cardiovascular risk, and those information was generated from clinical trials, but also from Mendelian randomization studies. And also what you have to focus here is that the effect of risk factors on cardiovascular outcomes is proportional to the magnitude of exposure and also the duration of risk exposure. So how much time you spend exposed to the risk factor. So the idea of that we've introduced this concept of cholesterol years, blood pressure year, or smoking pack years. So here is the first graph, which shows you that first you have to derive the trajectories for individuals. How is their cholesterol level over a lifetime? I've taken here the example of cholesterol level, but you can do with any risk factors. First, you calculate the mean LDL-C for each individual, and then you have calculate the mean cumulative LDL-C at any given age for each individual. And then the third step within the second category is to actually display number of scenarios where people have higher or low cholesterol level and the time of initiation of treatment. So if you initiate treatment at age 40 in comparison to age 40, what is the trajectory of cholesterol will look like over a lifetime? And the most important thing is the effect estimation of risk factor are incorporated into the model using the formula. So this is a risk which is adjusted for age and sex, specifically for cardiovascular disease. And RI is that one, whereas risk just purely R is age and sex specific rate of CVD for the whole population, whereas RI is specifically for individual patient. And then you have the measure of association from Mendelian randomization studies, which you can take from published sources, but you can also derive yourself as I pointed out before. And then the time weight between the mean cumulative level of risk factor for an individual into the simulation versus for the whole population. So this will give you this idea that for every millimole reduction in cholesterol over time, how much benefit we are going to be having by reducing cardiovascular disease. And this information can be only generated via Mendelian randomization because randomized clinical trials are not long enough to provide that lifetime impact on cardiovascular disease. And then once you derive that, then based on the scenarios that I've shown you before, you can look at different treatment options when you start early, when you start late, how would that translate into the risk of a myocardial infarction or stroke. And then I think that's the easy part, to design the microsimulation model and perform the health economic analysis. So the proof of concept for a microsimulation health economic model using Mendelian randomization studies, we first applied this technique in early 2018-19, when we actually looked at the cholesterol levels in children with genetic conditions such as familial hypercholesterolemia. And we've shown that inclusion of cholesterol years in this group of population matters. And what we've shown with this study is that if we start early at age 10, because these people have to be started according to the guidelines, then we actually save around 8 years in comparison to the standard of care. And then we applied this concept looking at specifically statin treatment. And then after a few iterations, we finally developed a model where it can look all potential risk factors for cardiovascular disease. So not only cholesterol level, but we looked at systolic blood pressure level, smoking, lipoprotein A, and the diabetes status. So what does the model does? It ages individuals between age 40 to 69, and we follow them up until age 85. This is a microsimulation model, and as I pointed out, that the risk event is proportional to cumulative LDL-C, systolic blood pressure, smoking, and diabetes. So risk of this event was directly taken from UK Biobank, but some of the utilities or quality of life data was taken from published sources. So the model functions in the way that we start as healthy, but individuals can have diabetes, can have MI and stroke. They can have both, die from cardiovascular causes or die from other causes. You can see here that we have included the two most common outcomes for CVD, because that's the bulk of evidence that comes from Mendelian randomization. We also looked at not only healthcare perspective, but also we looked at societal perspective in this analysis. And then you design and perform microsimulation model. And the way how the model function is that you iterate each individual. So each individual will have their own pathway during a 60-year time horizon. And then you design microsimulation and perform probabilistic sensitivity analysis for each individual. We've been very fortunate to use this model and publish in a number of very good, impactful journals, of which Padraig has been part of. Some of the most important things that we have been able to convey is look at the relationship of current clinical tools that we use for decision making versus if we would use lifelong decision tools. And what we've shown here is that if we would use immediate versus initiation of treatment much more early with a lifetime risk approach, it's going to be more cost effective rather than the current standard of care. We've also been very fortunate to work with a global team to look at the implication of lipoprotein testing for primary prevention of cardiovascular disease. And what we've shown here is that inclusion of lipoprotein A into a cardiovascular disease calculation matters and reclassify individuals and has a large impact on prevention of cardiovascular disease. So the impact of the model, to summarize, so model can simulate risk factor modification for any individual starting from age 30 to 84 with risk proportional to cumulative exposure, quantifying the concepts of cholesterol years, blood pressure years, and smoking pack years. So obviously using genetic variants to randomize individuals. The model can aid public health and clinical decision-making, especially when clinical trials for long-term primary CVD prevention are impractical or infeasible. We don't want to replace Mendelian randomization with clinical trials, but when clinical trials are not possible to undertake, it will be much more reasonable to do Mendelian randomization. We know that current guidelines use only 5-year risk estimation or 10 years, missed opportunity for prevention, especially in cardiovascular disease and potentially in dementia and in cancer. It evaluates cost-effectiveness, complex prevention, and identify individuals from whom long-term prevention is most beneficial. And our model is structured, it's not RCT, but can be applied to other contexts as well. This model also can incorporate distributional cost-effectiveness analysis. And we have made the model publicly available soon. The beta version of the model will become soon available. And we have included all possible therapies that are reimbursed within the Australia's pharmaceutical benefits scheme. And this can be tested, and you can actually check what is the most cost-effective way to treat individual with the risk of cardiovascular disease. Thank you for your attention. So this is how much I had for you today.
Thanks so much, Zanfina. That was excellent. Thank you. We'll hold off on asking questions now. We'll leave them at the end. Padraig, over to you now.
Thank you. Hopefully you can see that okay. Great, thank you. So this talk is going to cover a little bit of the same ground as Zanfina's excellent presentation did, but also go in a little bit of a different direction, talking about a new way to do long-term intervention cost-effectiveness simulation using Mendelian randomization, building on the types of evidence that Zanfina covered in her talk. So I'll just get straight into the presentation. So I'm thinking about a specific context here of SGLT2 inhibitors. So SGLT2 inhibitors are sodium glucose cotransporter 2 inhibitors. They are a drug used to treat type 2 diabetes. They work by removing excess glucose from the body. And there has been an increasing amount of evidence in recent years that they seem to reduce the risk of prostate cancer in men. So in some work with colleagues last year, we looked at this issue and we attempted to do a triangulation study. So we looked at electronic healthcare evidence, so a target trial emulation, looking at the association between these drugs and prostate cancer using medical record data. We also did Mendelian randomization, looking at the effect of genetic variants that mimic the effect of SGLT2 inhibitor drugs on prostate cancer. And then finally, a more conventional multivariable observational study looking at the association between HbA1c, so that's glycated hemoglobin, essentially blood sugar, and incident prostate cancer. And the reason for doing that last bit of analysis was to see if the effect of these drugs on prostate cancer risk operates through a glycemic channel, so essentially reducing the risk of prostate cancer, or is it through a non-glycemic channel, which would be potentially more interesting, because then if these drugs operate other than via blood sugar, that means that they could be repurposed and be useful for reducing the risk of prostate cancer in men who do not have type 2 diabetes and who do not have elevated blood sugar. So the simple logic here is that somehow by inhibiting SGLT2, this will reduce prostate cancer risk, which will reduce prostate cancer incidence. And indeed, in this triangulation study, that's exactly what was observed. So for a roughly 1.1% reduction in glycated hemoglobin levels, the odds of prostate cancer were reduced by 71%, so an odds ratio of 0.29 in Mendelian randomization in the main analysis, a reduction in the odds ratio of 49% in the replication analysis, and then in the conventional multivariable study, in the target trial emulation reduction in the hazard rate for prostate cancer. And importantly, this was also a non-glycemic channel. So that's raising the very intriguing possibility that we could have significant reductions in the risk of incident prostate cancer by repurposing these drugs, which are fairly well tolerated, which are widely used, very widely used actually amongst the diabetic population. So that raised the question, what should we do next? So we have about two sides of a triangle. We've got genetic evidence. So the SGLT2 genotype suggests that men who have this genotype have much lower risk of incident prostate cancer. The target trial emulation evidence also suggests that. What we don't have is RCT evidence. What we don't have is economic evidence. And if we set ourselves the question of exploring this from an economic perspective, we have different options available to us. We could run the RCT. The RCT will take a very long time to do because prostate cancer has a long latency period. You need to wait a long time for men to develop prostate cancer, for men to die from prostate cancer. So that trial would take a long time, although obviously it would be the best source of evidence. We could think about building a Markov model. We could think about the type of marker simulation model that Zanfina was presenting for all of the cardiovascular work that she led and is continuing to lead. And all of those would be useful and potentially valuable. One issue with the Markov model, marker simulation type model, is in themselves, those models don't give you some of the evidence that you need to populate those models. And that's exactly what Zanfina was talking about earlier. She created some of that evidence using the Mendelian randomization, then could use that evidence to populate the marker simulation model. And what I'm going to talk about in the rest of the presentation is both creating that evidence for prostate cancer and other cancers, and then doing an entirely new way of evaluating long-term intervention cost-effectiveness using Mendelian randomization in what's really quite a simple simulation. And the specific question in that simulation would be, imagine if we could give an SGLT2 inhibitor, so 300 mils of canagliflozin, which is widely used and fairly cheap SGLT2 inhibitor, to men over 50 to present incident prostate cancer as a prophylactic population-wide intervention. So this would just be almost like putting this drug in the drinking water. All men would get it, and then we'll see if it would be cost-effective to do that, bearing in mind that the evidence from the genetic Mendelian randomization study and the target trial emulation suggests very big impact on risk. We can't stop there. Obviously, we have to go and assess the cost effectiveness. So there are two aspects of this. One is to generate that causal long-term evidence that Zanfina was talking about, and the second was to do the intervention cost effectiveness. And I'm not arguing this should replace RCTs. I'm not arguing it should replace microsimulations or Markov models, but these Monte Carlo simulations can complement and very efficiently and quickly assess intervention cost effectiveness. And that can be a complement, or it could be to anticipate whether an RCT is even worth doing. So just thinking about the first part of that, and to very quickly revisit the assumptions, we want a variant instrumental variable that is related to our exposure X. We want the instrumental variable to be independent of confounding variables. And ideally, the instrumental variable will only influence the outcome via its effect on the exposure, not through this alpha channel at the bottom of the screen. So that would be a violation of the third assumption, a violation of the exclusion restriction. So the first three assumptions will detect the presence of an instrumental variable. With further assumptions, we can quantify the size of that effect, typically by assuming monotonicity, which would identify local average treatment effect, which we can define as the average causal effect amongst individuals whose value of the treatment was influenced by the value of the instrument. This is always worth interrogating, but broadly speaking, in Mendelian randomization, depending on where the data for the instrumental variable come from, the late is usually satisfied for most, if not all, of the population. So the parameter we want to estimate is beta. So that's the parameter going from our exposure, which in this case will be cancer, to an outcome, which will be cost, quality of life, quality adjusted life years. And we can estimate that parameter beta by calculating a simple ratio. So if we calculate the association between our instrumental variable G and the outcome, for J instruments, we can call that parameter big gamma J. The other part of the ratio that we need to calculate is the association between the instruments G and the exposure. We call that little gamma G. So if we estimate those in a simple regression, regress the exposure on the genes plus on the genetic instrumental variables on an error term, we regress the outcome on the same set of instrumental variables plus the exposure to account for possible violations of exclusion restriction and an error term. Do some simple algebra, and we calculate the ratio of big gamma J to little gamma J, and that boils down to this term. So alpha J over little gamma J plus beta. If alpha is 0, this first term in this expression will be 0, and therefore we'll have estimated the true parameter. Then the question is, under what circumstances will alpha J be 0? It'll be alpha J will be 0 if the exclusion restriction is satisfied, and there's no path that's going from the instrumental variables to the outcome except via the exposure. If there is such an effect, that is a violation of the exclusion restriction. That's known as a phenomenon in genetics called pleiotropy. Pleiotropy is where one single variant influences many different outcome phenotypes or many different exposures. So example might be particular SNP, G1 might influence prostate cancer and also depression. And therefore, when you measure or include that instrumental variable in your analysis, you're not measuring only the effect of prostate cancer on your outcome, but also these other things. And that's why it's extremely important to test very rigorously the exclusion restriction that alpha J is actually 0. In practice, pleiotropy tends to be very, very common. It's pervasive throughout the genome, and therefore it's very important to use methods that are robust to different types of pleiotropy, which I won't be presenting in this short presentation, but it's absolutely critical in Mendelian randomization that you interrogate this alpha J, that there aren't routes from your instrumental variables to the outcome that aren't via the exposure. So the next step is then to measure the exposures and outcomes. So big gamma J we get from UK Biobank again. So that's the association between genetics and the outcome. Little gamma J, we get those from genome-wide association studies. For this particular application, we wanted to have at least 2 genome-wide significant SNPs to include for our cancers. 8 cancers met those inclusion criteria: bladder, breast, colorectal, lung, multiple myeloma, ovarian, prostate, and thyroid. We got costs and qualities from UK Biobank. Costs in this case were inpatient and imputed primary care data. Qualities aren't reported directly in UK Biobank, so we calculated those using the Sullivan methodology, where we assign quality of life decrements to different conditions based on coding of those conditions in ICD-10 and ICD-9 codes. And the interpretation of the quality outcome that we measure is how qualities change by having versus not having the specific cancer over the average year of follow-up. Then thinking about this intervention, so if we imagine we had these data, how could we do the simulation where we're giving an SGLT2 inhibitor to all men over 50? So the hypothetical intervention we're imagining is assuming people got 300 mils of canagliflozin from their recruitment into UK Biobank cohort. Why 300 mils of this drug? Because that's very close to the reduction in HbA1c in the Zhang et al. Paper. And then the simulation is very simple. Identify the proportion of men who develop prostate cancer in our data, the proportion of men who would not have developed prostate cancer if they received the intervention. And then you compare the healthcare and quality of life, quality adjusted life years that differ between those 2 scenarios. The assumptions underlying this, the comparators do nothing, and that's a relevant comparator because there's nothing that is routinely offered to men as prophylaxis for prostate cancer. We assume all men in the model take their little pill every day, and we didn't model the side effects of SGLT2 inhibition. And the simulation is just to repeat the simulation 10,000 times. So going into that sort of core step, the three quantities in intervention simulation, you identify the proportion of men in UK Biobank who are diagnosed with prostate cancer in general and in different age bands, convert the odds ratio from Zhang into risk differences, and then finally, just as in Zanfina's presentation, you can use the Mendelian randomization results or conventional multivariable adjusted results, and then calculate net benefit to assess cost effectiveness. So it's that simple. There's three steps, none of which are desperately onerous, provided the data is there. So in terms of results, there were just under 311,000 individuals, unrelated individuals in UK Biobank, of whom 54% were female. Mean age of recruitment was just under 57 years, and we followed them up for a mean of 8 years. Mean total healthcare costs of just over £600, and breast and prostate cancer were the most common cancers, as you would expect. So this is the set of results for all cancers from Mendelian randomization, and a couple of things to draw your attention to. First is all the cost estimates are positive, which is what you'd expect, but most cancers have extremely wide confidence intervals. The narrowest confidence intervals, are, as you would expect, are for the most common cancers, which are breast and prostate cancer. For the others, the confidence intervals are extremely wide. Similar picture with QALYs. Again, breast and prostate have the most narrow confidence intervals, but again, the QALY impact is very negative. The results are imprecise for less common cancers, both because they're imprecise, but also very importantly, the proportion of variance that is explained by the SNPs for those cancers is very low, and the power of Mendelian randomization is a function of that variance explained. For some of these cancers, the SNPs explain very, very little of the variance, and therefore the confidence intervals are extremely wide, which is something you have to keep an eye on when you're interpreting your results. If the variance is low, you're going to have super wide confidence intervals. It's also worth bearing in mind that UK Biobank is full of healthy, wealthy, educated people who have better health behaviors than the general UK population from which they're drawn. So you can interpret the results in that light. Apart from the sex-specific cancers like prostate, there are no apparent effects from sex or pleiotropy, so no significant violations of the exclusion restriction. And in the paper, which describes this in more detail, you can see all of those results. Then, thinking about the results of the hypothetical population intervention, this is just simply an ICER calculation and a net monetary benefit calculation. We have healthcare costs, we have quality adjusted life years per year, and then you do the simple net monetary benefit calculation at a cost effectiveness threshold of £20,000, and this is highly negative. So it isn't cost effective despite a 71% reduction in the odds of developing incident prostate cancer. It's not cost effective. The drug would be cost effective if it costs about £1.20 per month. So if it was the same cost as a generic statin, it would be cost effective. Which is interesting because there's such a huge reduction in the risk of prostate cancer. But a feature of prostate cancer is it doesn't have absolutely drastic impacts on quality of life. It doesn't have drastic impacts on healthcare cost. But it so happens that this is the application we use because there's so much interest in SGLT2 inhibition as a way of reducing the risk of prostate cancer. So I think in this case, it shows it's worth doing this very quick and simple simulation using Mendelian randomization, because even though the intervention appears tremendously attractive from a risk perspective, it's unlikely to be cost effective based on this evidence, which we can use to complement the other sources of evidence that we might generate, such as a microsimulation or in time an RCT of this drug. So is this a sensible thing to do? So there are clearly many, many assumptions, and always what you're measuring in Mendelian randomization is a genetic liability to the exposure or to the particular risk factor you're studying, which means that the stable unit treatment value assumption is violated. So what we've simulated or what we've imagined is a cohort of men who are getting prostate cancer because of their genetic risk. We don't really know if that risk is materially different from the risk you get from non-genetic sources. In some cases in Mendelian randomization, we can be quite confident that that risk is exactly the same as non-genetic sources. In other cases, it's essentially impossible, or there isn't a sensible way to characterize that. We can also get dynastic biases in Mendelian randomization. That's where people with genetically similar profiles may have children. This is probably unlikely. It's unlikely that people choose their partners based on cancer risk. If you're looking at an exposure like education or smoking or consumption of alcohol, that becomes much more relevant. Within family studies are becoming, or have become actually, the gold standard within Mendelian randomization, because you get controlling of the local family environment, which helps isolate the most relevant genetic effects. We don't have that for these kind of exposures yet, primarily because those cohorts don't exist, but also statistical power is an issue, and that would compound the challenges that we saw with statistical power. However, I think it does demonstrate that we can very quickly and efficiently do this type of analysis to assess long-term intervention cost effectiveness. And touching on, again, one of Zanfina's themes, often the thing we really care about is long-term impacts, not necessarily the acute impact. And that's what this is measuring. So again, it's something that can complement, but not replace these other types of analysis that we conventionally do in health economics to assess intervention cost effectiveness. I missed something, go back. So just final slide. It's efficient. I think you need to bear in mind the explanatory power of any SNPs that you're using in your analysis. That's why the confidence intervals for those less common cancers are so extremely wide, essentially prohibited inference. Also think about selection. UK Biobank is very, very, very heavily used, but it's very well appreciated now. It's healthy, wealthy, not representative cohort. However, despite all of that, we did see material impacts of prostate cancer and breast cancer. Then going into the simulation of prostate cancer, specifically using SGLT2 inhibition, bearing in mind what we really care about in cost effectiveness analysis are essentially three parameters: the thresholds, and then cost impacts and quality impacts. We can estimate those directly in Mendelian randomization, and then do that simple Monte Carlo simulation to assess long-term impacts. In this case, we don't know what the price would be for a novel indication of SGLT2 inhibition in prostate cancer. I assumed it would be the same prices for type 2 diabetes, but that, of course, would need to be seen. And then finally, I think this wouldn't take away from the case for doing an RCT of SGLT2 inhibition. But of course, we do have to be a little bit sanguine about the prospects of it being cost effective. So I quickly thank my colleagues, Sean Harrison and Richard Martin, UK Biobank Resource, and very kind funding from Cancer Research UK. And if you'd like to read the paper, it's in Medical Decision Making. And I will stop there. Thank you.
Wonderful. Thank you very much, Padraig, and well done to both of you. Fantastic presentations and wonderful insights. Let's open it up now for discussion. If you have a question, please raise your hand and I'll facilitate the discussion.
So, Elias, I think there is a question from Dante in the chat about testing for gene environment equivalence, which is a really important point. We generally can test it directly, but there has been a huge exponential growth in the volume of Mendelian randomization papers, and a lot of them have become really low quality. And one of the ways in which they are low quality is they're not sensibly interpreting gene environment equivalence. What you want ideally is a justification of why the genetic variant is a reasonable way of instrumenting your exposure. So there are lots of papers looking at air pollution and doing GWASs of air pollution, but many people would argue that's not a sensible there isn't a sensible gene environment equivalence in that case, because what's the corresponding biological process in the body for air pollution? It just doesn't exist. So it's much better to restrict Mendelian randomization where there is an internal biological process in humans that can be sensibly instrumented using a genetic variant. And that's where a lot of this literature falls down, is looking at things that just couldn't possibly or plausibly be instrumented using genetics. And at that point, you should stop going any further and not pursue the Mendelian randomization. So you have to justify that. You need biological processes influenced by genetic variation, not things like air pollution or the type of what are other examples? Maybe some types of food that you might eat aren't really sensibly instrumentable.
Yeah, I agree, Padraig, with your response. We have to focus on evidence first that we've generated from randomized clinical trials, and then things that are supported by Mendelian randomization as well. So we need a very pathophysiological and biological evaluation of these risk factors with an outcome. And some of those gene environment studies are not as good as those that are related with common disease outcomes that we are assessing today.
And then Dante has a, I guess, follow-up questioning about gene environment assumptions. No, I don't think I've ever explicitly been questioned about that. But I think I would want, if it was an unusual exposure that didn't clearly relate to biological processes, it really does require a lot of justification. And I think the best Mendelian randomization studies have been in the area that Zanfina has been working on, where you have proteins that can be measured very well that are circulating in the body. There's a lot of, imperfect, but a lot of very good biological knowledge about what they do, where they come from, how they operate. And those are very, very heavily and directly influenced by genetic variants that are understood. As we move further away to things like education, things that are more biologically distal, it becomes a little bit more challenging, but I think it's still sensible. But once you get too far removed from a connection to biology, then there are problems.
Thanks, Padraig. My question to you would be, what challenges you see about the use of Mendelian randomization within HTA, and if there is anything we can do as a discipline to support the use of Mendelian randomization or other real-world analysis methods.
I'll let Zanfina go first on that one.
Yeah, I guess for me, working in cardiovascular space, the most challenging thing is, first of all, if you do your estimates yourself, it's different. But then when you use the estimates from published sources, as Padraig pointed out, which one to use, which is the most reliable estimate, because we have different estimates for a single variant from different biobanks. It's not just UK Biobank, but there are other biobanks available as well. That's challenge number one. What is challenge at the moment, because not many researchers are working in this field, is that to use some kind of a checklist to make sure that we have ticked everything, that when we publish such studies, Mendelian randomization and health economics, then people who evaluate those studies are confident that what we have produced is the highest evidence available at this point in time. So the challenge is creating this checklist. We can use the Mendelian randomization checklists that are used in epidemiological research. That's a very good start. But then when it comes to HTA and then linking the quality of life and costs, I feel like we need a different checklist. So this might be one of the key challenges. And I think the second challenge that I see is that Mendelian randomizations are mentioned in the NICE guidelines, but I don't see them being mentioned in Australian guidelines or Canadian guidelines to be used in a real-world evidence. And I think our challenge is going to be convincing government that this type of evidence is as reliable as Mendelian randomization evidence.
Yeah, I think it's definitely the case that Mendelian randomization is very much in its infancy as a source of evidence and as a tool that could be used for HTA. So with some colleagues at Oxford and NICE, we wrote a paper this year looking at NICE decisions, and I think we found one technology appraisal citing Mendelian randomization evidence, which is encouraging, but I think we still need a lot more thinking about where and how exactly it can be useful. I think it can't do everything. It would be wrong to oversell what it can do, but I think for those long-term impacts for complementing other sources of evidence, for also bearing in mind usually what we're interested in is the causal effect of an intervention. When we look at, say, a Markov model where you're looking at a state and you're putting a cost or a quality impact on the state, usually those pieces of evidence come from confounded observational studies as opposed to causal studies. So there is, I think, a lot of scope for it to become useful, but there are some well appreciated and some less well appreciated complexities, and it's understandable, I think, that reimbursement bodies, HTA bodies, will be a little bit cautious about suddenly diving into this new area. But I think it's something they should think about more, because it can add a lot of value, I think, to what they do, without too much pain, I think.
Awesome. Okay, well, thank you very much for your presentations. It was a pleasure to have you both here. And thanks to everyone who attended. It's really nice to see those events being well attended. So thank you very much. So before I close, a couple of things to say to all our membership. Can you see the slides? I'm sort of coming through.
Yes.
All right. So
Yes.
we have a few more sort of webinars and talks that are coming. One's specifically focused on low and middle income countries, and the other is on early career researchers. If you're interested in participating in any of those, please reach out to our conveners. You have the emails here. Zanfina is also a convener for LMIC talks. Please reach out to us. We want to support that those webinars too. And also, please engage with us. We have the LinkedIn page, the Blue Sky, and Twitter. Please stay in touch with us and learn about the activities that we do. And with that, so I'll draw this to close. Thank you very much, Zanfina. Thank you very much, Padraig. It was awesome. Thanks, everyone.
Thank you for the invitation. Thank you.